Hello everybody. This week's lecture is more about attention and then the visual depth perception. So we'll mainly talk about those two things. In last lecture I covered a part of attention. Uh, Today we'll talk more about attention. Then the second part of today's lecture, I'll talk about the visual depth perception. And next week, we'll talk about the plasticity of the sensory system. So, two brain areas are more involved in attention related activity. One is the posterior parietal cortex. So the posterior parietal cortex is one area that is heavily involved in attention, and then further higher area is prefrontal area where you have the frontal eye field, FEF here. So these are two areas where most of the attentional tasks or attentional cues or activities are processed. Now you see here if any lesion occurs in the posterior parietal areas, then there is a clinical term that is called unilateral visual neglect. So unilateral visual neglect can occur due to lesion of the posterior parietal area in one side. Okay. Here you see the behavioral responses by people with left sides visual neglect. That means this patient had damage to the right posterior parietal cortex. Okay, so he suffered from unilateral visual neglect on the left side. That is, he had difficulty in attending to the left visual field. In A, you see here, the subject, the patient, was asked to cross the lines and just put a mark on the lines, and he was given unlimited time to complete, putting a line or cross on those lines. And after he finished his tasks, you can see that what he did is he crossed or marked lines on the lines of the right side of his visual field. Okay, and left side he totally neglected. He did not pay attention and did not perform any tasks in the left side left unattended. So that is the deficit of function in one hemifield. And this is another task he was given. Uh, he was told to place mark at the center of the line. So in each case, you see the patient placed mark too far to the right. So that indicates that the left end of the line was neglected. So he was told, this is another patient, not this patient, different patient. So he was told to put a line in the center of the line, put a mark in the center of the line, but he was biased towards the right. So he neglected a part of the left side of his visual field. And this is another patient. So these are from three different patients. All had lesion in the right posterior parietal cortex. And here he was asked to draw a flower in a vase. And you can see that he was asked to draw from his memory a flower in a pot, right? And you see that he omitted uh, elements on the left side. So he didn't complete drawing this side. So part of this or left undone. So you can see that what can happen due to lesion in the parietal posterior parietal cortex. That clearly indicates that posterior parietal cortex is heavily involved in attention, visual attention. Okay, now we'll talk about the an experiment on the monkey's posterior parietal cortex. This should be actually posterior parietal cortex. Okay, so the recording was performed from the neuron of the posterior parietal cortex. So this is the neuron, and this is the firing here of the neuron, and this is the receptive field of the neuron, this one, receptive field. And this broad arrowhead line, blue line, is showing the direction of attention. So this is important. You can see the change of direction of attention by this one. And fixation point always appears in the center of the screen, where the monkey has to fixate his eyes, maintain fixation. Okay, and the direction of gaze is this one, and visual stimulus that appears inside the receptive field. Okay, and this is the neuron. Okay, so in this box here, you see that monkey fixates eyes on the fixation point in the center. Okay, in that dark point, and this is the receptive field of that recorded neuron here, and a stimulus appeared inside the receptive field. You see that red bar, and however the monkey was trained not to pay attention to the target that appeared inside the receptive field. Monkey, during the entire period, the monkey had to keep paying attention to the fixation point. And you see here the fixation point color changed. It uh, becomes dim. Okay, so dim, and then when the color of the fixation point changes, then the monkey uh, had to move the hand and touch the place where the fixation where the target is. So he moves his hand and with his finger touches the computer screen on the target. However, during this entire period, monkey didn't pay attention to the target inside the receptive field. So here you see of no change of the firing, no attentional task, no increase of firing of the posterior parietal neuron. Uh, then inside this box, this is a second task. Monkey has to move the eyes. Suddenly, monkey will move the eyes from the fixation point to the target inside the receptive field. So here, same thing you see, monkey fixated eyes here in the center, and firing here spontaneous. Then monkey, uh, in this case, at time 2, same as this one, right? A target appeared inside the receptive field, and now monkey, in this task, monkey has to pay attention, okay, to that target that appeared. So you see the attention showed by this one, so that has been moved towards the target, okay. And fixation point disappeared here, okay. So after fixation point disappeared, the monkey had to pay attention to the target and then shift the eyes to the target. So you see, when the monkey was paying attention to the target, before it moves uh, his eyes to the target, the firing increased here, and the firing continued, increased during the movement of the eye. So we know that posterior parietal cortex processes the attention as well as uh, intended movement. So this is what you see here during attention as well as intended movement, grasping or moving the eyes. Uh, the firing increased, and in this box here, first two steps are same. Monkey fixated, uh, receptive field is here, target appeared inside the receptive field, monkey still continued fixating, okay. Then a target appeared, so monkey changed the shifted the attention or changed the attention, right, from the fixation to the target. See this one, and the firing increased, okay. So when the monkey was paying attention to the target, the firing was high, response was high, and then the monkey had to move the hand to the target and touch the screen, okay. And during this intended reaching movement, the firing was also high. So you can see that posterior parietal neuron not only processes attention, but also attention um, uh, cued task, uh, movement of the eye, intended movement, or grasping or reaching movement. All those functions are heavily processed in the posterior parietal cortex. This is a very similar experiment on the monkey. Posterior parietal cortex neurons were recorded, and monkey performed different tasks: fixation task, saccade task, reach touch, and peripheral attention touch. And you see the firing was high during the shift of the attention from the fixation to the target, and movement of the hand, and here while the monkey was waiting to see the fixation point dims. Uh, so here the firing is not high. Okay, so only in these two cases you see the firing was high. Okay, here you see the firing increased briefly for a brief time. That is due to the change of the fixation point, not paying attention to the target, but monkey was paying attention to the fixation point, and the color of the fixation point changed. It became dim. So that triggered some response, but it did not continue because he was not maintained the attention task. So this is actually very similar to the last one. Now, attention and human brain activity, fMRI recording in the human brain. Okay, and in this case, the subject, human subject, was given the task to shift the attention from the right visual field to the left visual field. Okay, and what he was instructed is uh, first, for example, he was asked to pay attention to the right visual field here, and when the letter appears here, L, that is the indication or instruction, visual instruction to the subject that now you have to shift your attention to the left. Okay, and when he was maintaining attention in the left side, a letter R appeared here. That is the instruction for him to shift his attention to the right side. Okay, now they recorded the activity of the posterior parietal brain activity, and they found that when the subject was paying attention to the left visual field and was waiting to see R, the activity increased in posterior parietal cortex, shown by this orange or yellow color. And the activity, increased activity of the left hemisphere was shown by the blue color, and the activity was high when the subject was paying attention to the right visual field but was waiting to see the letter L to shift to the other side. Okay. So here you see the yellow line is showing uh, the activity, increased activity uh, of the right hemisphere. Okay, and the green line is showing the activity of the left hemisphere. Okay, so the activity was high when the uh, subject was paying attention to uh, a visual field of the opposite side, because we know that the signal from the right visual field goes to the left hemisphere and left to the right mostly. So when the activity in this side increased, the activity in this side decreased. So you can subtract and see the difference. And when the activity in this side increased, the activity of this side was suppressed. That's what you see here. So this is also showing that the activity of posterior parietal cortex changes a lot during attentional task. Uh, awareness and attention. Awareness is active thinking about or concentration on some source of stimulation. Okay, so awareness is not attention. It is before you pay attention. For example, you are waiting in a bush for hunting, and you are you know that in certain part of that area you are expecting a tiger or something will appear. So you are sitting here and you are paying attention to this area, not other areas only. You are paying attention, uh, but this is not attention. You are that is the awareness. Actually you are expecting something to appear here. You're thinking that something might appear in this area. So that is the awareness. That is not really attention. It is before attention. Now, if you see something, you see a tiger has appeared here. Okay, then what you will do? You will pay attention to that target. So that is the target now, and you will make sure that what is that? Look into more detail of that object right inside the bush to make sure that it is your target. So that is attention. So awareness that something will might appear or something might happen in this area, that is the awareness. Like if I tell you that pay attention to the upper right quadrant of the screen, something will appear and you have to detect that. So before that appears, that object appears, that is the awareness. You are thinking that something will come here, and when the target appears, you don't know what that target is. You have to identify it. So when the target appears, you pay attention, see its shape, size, feature, other features to identify the object. Attentional cueing. Many ways we can give cues. In last class I talked about that. You can use simple an arrow that something will appear in this side, right? So that is a cue. If I put like this, that is a cue that something you will expect in this side. If no arrow, that means no cue is here, right? It could be in this side or this side. So that is neutral cue, and these are valid cue. Now invalid cue is if it appears like this. So you are expecting something here, but it appears in the other side. So that is invalid Q. Q is there, but that is invalid. Okay, so that is different than a neutral Q. And a distractor is another thing that can reduce your performance uh, to detect something while you are paying attention. So if the background is the similar color or same color, then that would be considered as a distractor. So the contrast is another thing. If contrast is more, less distraction. If contrast is less, then detection will be poor. So attention, the selection of some source of sensory stimulation for increased cognitive processes. This is important. Why we need to pay attention? We pay attention to increase our cognitive processing, to identify the object, to see the features, individual features of that object, right? Uh, so we pay attention to improve our or increase our cognitive processing, um, to memorize better. If you pay attention more, you memorize better and longer time. So memory is another, improving the memory, retention of memory, learning. All those things are directly related to attention, right? That's why we always say pay attention. More attention you'll pay, you will remember better, you will remember longer, and you learn better. Selective attention, very specifically, if you pay attention to something uh, and ignore other things. Okay, like in a big crowd, when you are talking to somebody, if you hear your name, somebody mentions your name in the crowd, then your attention shifts there, and you try to only focus uh, on that voice. So that is selective attention. For selective visual attention, you need to look at the object directly, and the signal should be processed in the center of the retina. That means where you have the cones more to detect the fine details of the object. Visual acuity is high. Filter theory of attention. When you increase attention to a specific thing, then other things become background. Okay, other things become noise or fuzzy. So that is the filter theory of attention. Other things are discarded. So we already talked about the valid, invalid, neutral, Q, and in last class we talked about this. This is one simple experiment. The subject has to fixate eyes, look at the center of the uh, screen, and then three types of cues appear randomly. If the cue is this one, that means something you should pay attention to the left. So the target will appear in the left side, and if it is a plus sign, that means no signal is there. This is a neutral cue. Okay, and this is also valid cue. And sometimes the subject was given invalid cue, like the arrow appeared this way but the target appeared here. So in 20% trials they used invalid cue. Here the arrow appeared in 20% cases in that way, but the target appeared here. So definitely in this case, in case of valid cue, the performance will be very high, like will be 100% correct, right? If someone pays attention, you know that the target will appear here, so you will respond quickly. The target is here and the reaction time will be short. Now if it is neutral, reaction time will be slower than the valid cue condition. And if in case of invalid cues, the reaction time will be longer because you are expecting it appear in one side, but it appeared in the other side. So in that case, the reaction time will be longer. You see, invalid cue reaction time is much longer than the neutral cue, and in case of valid cue, it is really short. So response is fast. Also, the correct response will be more in this case. In this case, the subject will miss, uh, correct response. This is another experimental design that is called Posner Cuing paradigm. In Posner Cue paradigm, it is actually very similar to the one I mentioned in last slide. The cue, the target stimulus is preceded by the cue. So first the cue will appear. You see here the subject will see first three squares or boxes, then one will light up, and then that is the cue that you would expect something to appear here. And if the object appears here, then that is the valid cue. And in this way you can also create invalid cue, like if this square lights up but target appears here, that is invalid. And if this one, it might appear here or here, so that is neutral cue. Visual search paradigm. We always use our search paradigm, or we always while you are driving or walking or doing something, you look around, right? And while you are driving, you look at the distance of the other cars, right? And you find your path, how to when you walk or drive in a crowd, how you find your way. So all those are involved, heavily involved in search, visual search. So the subject looks for a designated target in terms item among a number of distracting items. Okay, among other things, you try to find the way or desired object. So here, this is a simple search technique. If all other objects have one color, same color, and the object you are looking for, that one has a different color, then that's a very simple search technique, right? So you can quickly detect that. So this is in the lab experiments. The subject fixes its eyes here, and in the peripheral view, uh, different shaped different shape appeared. Some are L in different orientations, and also T. So if the subject is told that you have to just pay attention to the object that has a different color, this is a very simple task. You can quickly do that because you are not caring the shape of other objects. You are just only looking one thing, the color. Okay. uh, However, uh, if now the subject is told to find out where the T is, then it is a little more difficult than this one, because the color of other objects is same, only the shape is different, right? So you have to look at two things, right? You have to look at the orientation and shape, and since the color is same, it is a little bit uh, difficult. Uh, so using different stimuli, we can change the difficulty of the task. This is a relatively weak task, both, but you can make the task more difficult and measure the onset and reaction time. So here you see the reaction time in this way, and the number of object. So by changing the number of object, like if you are asked to find T when there are 20 objects, then if you increase the number of object, then uh, it will be uh, uh, more difficult. So here 10 objects, 20 objects, 30 objects, 40 objects. Okay. If in presence of all different numbers of objects, the subject can detect quickly, that is most efficient. And this is very inefficient performance of the subject. And that efficiency of performance depends on the type of task you are giving. In this task, like the subject has to identify the white among black. It doesn't matter if you give 20 other objects or 40 or 50, the time will take same amount of time will take, right? This is a little more difficult attention task. This one is called feature search. The most easy task, if you are asked that is the object that you are asked to identify is there or not. Okay, so quickly you can tell yes by looking at the color. You are only looking at the color. Here this is the target, so you have to see the orientation. as well as the color, because there are two different colors, red and blue, and two different, no, here you have only one orientation. So this is harder than this, but because you have two different colors, here other colors are same, red, here you have two different colors, so this is more difficult than this one. And these two are called conjunction search. So you have to see the color, and here you have to see the color as well as orientation. So color must be blue, and also orientation is horizontal, and you have both blue and red colors as well as vertical and horizontal, you know, uh, horizontal objects around that. So this is more difficult here. So in this case the response time will be very fast, here more difficult, and here further difficult. Inattentional blindness. This is something interesting, that while you pay attention to something, other things get less attended, or become background. So here you see the subject, human subject, was asked to fixate eyes on the fixation point here, and the a cross uh, object appeared after the subject fixated the eyes in the upper left side, and the subject was told that you have to uh, watch and notice which bar is getting smaller or shorter or longer. So the one of these two bars, vertical or horizontal, will change the size, length. So now the subject is paying a lot of attention on this one, right? Because he has to report that which bar changed its length. And when the subject was paying attention to this big cross, what happened here, you see, the shape of the fixation point changed. Okay, and after the task, the subject was asked, did you notice any change in the fixation point, shape of the fixation point? The subject was reported negative. The subject did not pay attention and did not notice that while he was paying attention here, the shape of the fixation point changed. Okay, so that is called inattentional blindness. This is the subject was blind here. He didn't notice the change of the fixation point, although his eyes were fixated on it. uh, Spatial frequency or contrast is another very important factor that can change your attention performance as well as your learning and memory. And if the contrast is high, then the detection ability will be much better. So the subject will be able to find more fine detail of within the image uh, and that's what you can see here. You can see a lot of details here. uh, See the writings, the monogram here or logo, whatever, you can see clearly because of that contrast. Here the contrast is almost no contrast, very weak here, so you cannot read any letter, any can't see anything clearly. It's very invisible. Although here the background and the object, the contrast is low, but the contrast is very high here and here, the center. Here, although the background and the object contrast is high, that's why you can see the object more clearly or more quickly. However, since the contrast between the color of the cup and the writing uh, is very low, that's why you cannot read or see the things. Binding problem. You already know that many different types of visual cues are processed in different areas of the visual part of the brain, right? Like your color is processed separately, your orientation is processed in a different area or areas. Color is processed in different areas. Shape is processed in another area. For example, orientation, uh, age, those are processed in the early visual areas. For example, your primary visual cortex, simple neurons, right, uh, can detect the age and also orientation. However, color is more processed in V4. We talked about that in last class. Uh, and the shape is processed in the inferior temporal or in the ventral stream, you know that. So all those cues give you the picture of complete picture of uh, anything. So here you see this is a complex visual environment because you have the crayons of different colors and different orientations. It's not highly complex, but it's kind of complex. So it is there. You see different colors and different orientations, right? So if you are asked about the orange crayon, then you have to use those brain areas. You don't do that, but those brain areas are used to detect the orientation, the shape, the color, the location, right, which is a function of location, which is a function of dorsal stem, parietal. So all those areas are used uh, to do that. So that is the binding. Different areas work finally put them together, the information. However, in more complex environment, the binding becomes more difficult, right? And that is the binding problem. In that case, the error will be more. Difficulty will be more. Chains blindness. This term is used to indicate the inability to quickly detect changes in the complex scene. And in most of the cases, our brain is not very good to detect the change in a complex environment, and that is called change blindness. Here you see a mosque, and in front of the mosque you see stall, and few people are there, and in the mosque there are different features, right? So if you are asked to see this one for a while, and then you pay attention to this one, this picture, and ask, do you see any change? It is not very easy to quickly detect that change in that changed image. So that is the change blindness. Here you see one window I can see is missing here. Okay, it was here. So it will take a while, some time, or you have to, if you don't go back to see that previous picture, you won't be able to answer at all. So that is the uh, term or condition that is called change blindness. Dichotic listening. This is something interesting, that your both hemispheres sometimes cannot pay attention simultaneously. So that is a weakness in our sensory processing, that sometimes the brain cannot detect or process two different things simultaneously or together. And this is a good example of that. uh, if you are presented two different uh, sentences, you are listening to different uh, sentences, one in your left and another in your right ear through the speaker, head speaker. So in the right side, he listened. Business people share ideas and encourage one another, but each business dot dot dot. And in the left ears, at the same time, you listen. Uh, Vegetables contain a variety of nutrients that can contribute to healthy. Now, after the subject listened both at the same time, when the subject was asked that what did you listen, then the subject will mention one of these two. The other, his brain completely ignored, and that is called shadowing. This listening is called dichotic listening. You are listening to different things in two different ears. That is dichotic listening, and uh, your brain is masking one of those two, and that is the shadowing. Uh, so that also happens in case of visual, not only auditory. Your visual brain is also, uh, you know, uh, not good to process two different things when they are presented in two different eyes separately. How a distractor can reduce the discrimination, the ability to discriminate or identify the objects. Here you see a number of horses. There are few, uh, and the background, like you see the snow and ground, the colors are very similar. So in that case, it's difficult for the subject to uh, identify the objects. So that is, we'll say the background is a distractor. Uh, this is one kind of visual illusion uh, that tells that your brain cannot see two things at the same time if they are presented in a particular way, which is also called winners take all. Winner takes all. So here, if you pay attention to this picture, uh, some people will say when they are asked what do you see, they will say I see a vase, flower vase. Uh, uh, Other people will say that they see two faces. You can see both, but if you pay attention to this one, these two become background. If you pay attention to the faces, then this part become background. Some painters, they use that trick and successfully did trick the brain. Here the kissing girls are two lover embracing. That's the name of that painting or work. uh, and if you pay attention to one face, then another becomes background. However, the painters can also uh, draw the things in a way that two things you can see simultaneously. So here it's difficult. One will be dominated than the other, but here both are dominated and you can simultaneously see both. Uh, like dichotic listening and shadowing, also your visual system uh, behaves very similarly, and that is called binocular rivalry. That means you present two different things in two visual fields or two eyes, then a conflict occurs in the brain. That is called binocular rivalry or perceptual bistability. So what will happen here, you see in this picture, two things are here: a girl face and a house. Now if you pay attention for a while, concentrate on that, you'll see sometimes the face becomes prominent, house becomes background, so you can only see the face clearly. And then uh, you will see that house sometimes become uh, prominent and clear, face becomes the background. So that kind of shadowing also occurs in the visual system. One side of the brain wins and then another side wins, so the shifting occurs. So this is an experiment. The recording was performed by fMRI in human subject, and the subject was shown this picture and he was asked to pay attention to the face. So when the subject was paying attention to the face became clear, prominent, came forward, and house became the background. And then the subject was verbally told that now pay attention to the house. So the house slowly appears and face disappears. And they recorded these two areas. You already know that parahippocampal place area that processes big landscape or house, those kind of structures, and fusiform face area processes the face. So you see the activity of those areas also changes. When the subject was asked to pay attention to the face, face becomes clear and the activity of fusiform face area increased, you see here. And the activity of uh, the parahippocampal place area was weak. And then when the subject was paying attention to the house, we see that the uh, parahippocampal place area activity increased in some occasions. Here you see more clear, okay, and face area is weaker. So that is a nice experiment on human subject. And both areas, you know, are located the lower part of the temporal lobe, inferior temporal area. Prosopagnosia is a clinical condition in which the subject has inability to recognize face. Specifically, the person has difficulty in recognizing face, but he can recognize the other objects. So this could be due to lesion in the fusiform face area, uh, particularly in that area if any lesion occurs. And topographic agnosia is a condition in which the subject can recognize the face but is unable to recognize spatial layout of landscape or building, street, and so on. So this is more due to lesion in the parahippocampal area, parahippocampal place area. However, in most of the cases when stroke occurs or injury occurs, it occurs in a larger area, and these higher areas process multiple things, as you know already. So we see the complications, other complications are also present. Sometimes we see the combination of two or more problems. Neurons of neurons of premotor cortex in front of the primary motor cortex have visual receptive field that responds to the visual stimuli near the hand. So that visual receptive field is around the hand, and uh, that receptive field size can be changed. It depends on our experience, how we use our hand in our tasks. So different people use the hands in different types of tasks. So that receptive field is constructed based on that. So this is one kind of plasticity, and that receptive field size can be changed by uh, changing the type of work you do using your hand. So here you see the recording was performed uh, and from the premotor area, and the receptive field of the recorded premotor area was here around the hand. So this is very interesting that you have premotor neurons, those neurons have the receptive field around your hand. So you move your hand, the receptive field moves with the hand. So it is always attached to the hand. And this is the receptive field size before the monkey was trained to use tool. And then the monkey was trained to use the tool to perform the task. This is the tool, extended hand, we can say. And now his visual coordination changes, right, to perform the task, as well as the motor coordination changes. And interestingly, now when his hand was extended, then the receptive field size of that premotor neuron also changed, got elongated. So this is a very interesting finding. Mirror neurons. These days you probably hear a lot about the mirror neurons. Uh, there are neurons in your brain, particularly in an area that is called area F5, and in the premotor cortex, area F5. Those neurons responds in multiple things that indicates the same action. For example, if the monkey watches that someone is breaking a peanut, okay, when the monkey see that the experimenter is breaking a peanut, the neuron responds by showing high number of action potential or spikes. Okay, so this is one condition. Now when the monkey is given the peanut and he breaks the peanut, same neuron increases the firing. So same neuron is processing those two. Not only that, even when the monkey is not doing or seeing someone breaking the peanut, if the monkey listens that sound, the same neuron responds. So that is the property or characteristics of um, a mirror neuron. So it responds to multiple modalities, although those modalities are the meaning of those modalities is same. It is like more like grandma neurons. We know that we say grandma neurons, that you when you are small, you used to spend time with your grandma. So if you hear, like after her death, if you hear someone's voice uh, like your grandma, the neurons fire in the brain. And when you see someone dressed up like your grandma, same neurons fire. If you smell your grandma, same neurons fire. So all those things are giving you the feeling of your grandma. So very similar uh, type of behavior in mirror neurons. Here you can see the recording was performed from monkey's mirror neurons in F5. Monkey reached to pick up food with fingers, so the neuron responded strongly, and monkey watches human or experimenter reach to pick up food with finger, response of the same neuron is high. And when the monkey watch human reach to pick up food with tool, in this case the response is much weaker. So using hand and doing same task, that triggered the response more. And the mirror neurons, as I told you, even if you hear the sound of the same task, also responds. So responds to multiple actions, and the mirror neurons have been, recently studied a lot, and that study uh, linked the mirror neurons to a common clinical, condition that is called autism, uh, autistic spectrum. Multiple things go on in the autistic people's brain, and uh, the lack of mirror neurons have been linked to the autism, or at least some type of autism, not if not all. That when you see that you see someone is in trouble, you feel emotional, right? When you uh, see someone is getting a shot, injection, you feel that kind of painful, emotional thing appears in your mind. So because of the mirror neurons, experimenters or scientists have indicated in a number of experiments that uh, those are due to the uh, mirror neurons in your brain. You see and feel emotional when you do something, or another person does similar thing. Okay, and if now think that someone's brain has lack of mirror neuron, then that person will not have enough emotion, will not feel the pain or joy, right, of another person, will be less sympathized, or less emotional. Empathy will be less. So we see those things are present in some kind of autism. So although experiments are still going on, and autism is an spectrum, so multiple areas could be also involved in it. So let's stop here. In next lecture, we'll talk about the depth perception, visual depth perception. Thank you.